It is well established that parasympathetic stimulation of muscarinic receptors is involved in both normal and overactive bladder muscle contraction. Muscarinic receptor subtypes are located with overlapping distribution throughout the body. To date, there are 5 identified subtypes with varying functions. For example, M1 are highly expressed in the brain and have a critical role in memory and cognition. And in the salivary glands, M1 receptors have a role in mucus secretion. M2 receptors are present in the heart, mediating parasympathetically driven bradycardia, and in the bladder, where their role is unknown. M4 are present in the brain, where they may have an analgesic role. The M5 receptors are also present in the brain, where they mediate dilation of cerebral blood vessels, and in the eye's ciliary muscle, where they mediate contraction. M3 receptors are present in the brain, eye, salivary glands, GI tract, and bladder. Both M2 and M3 receptors exist throughout the bladder wall. Although M2 receptors outnumber M3 by a 3-to-1 ratio, it is the M3 subtype that is the primary mediator of detrusor contraction. Let's take a closer look. The layers of the bladder wall include the smooth muscle bundles of the detrusor muscle, the lamina propria, and the urothelium. Parasympathetic fibers provide excitatory input to the detrusor muscle. A delta and C afferent fibers return sensory information. On average, the bladder stores 350 to 500 milliliters of urine. When about 200 milliliters of urine accumulate, the first sensation of an urge to urinate occurs. As the bladder fills, the wall stretches, stimulating afferent nerve fibers. Afferent signals travel to the brain, where they signal the urge to urinate. These signals contribute to activation of the micturition reflex. This reflex sends efferent signals via parasympathetic nerves to the detrusor muscle to cause contraction. Let's take a closer look at the mechanism. At the neuromuscular junction, the nerve impulse stimulates release of acetylcholine, which binds to M3 receptors, the primary mediators of detrusor contractility. The M3 receptors then signal the release of calcium from intracellular stores to initiate contraction of the detrusor muscle for normal voiding. In normal bladder function, the detrusor muscle relaxes as the bladder fills and contracts as it voids. However, in patients with overactive bladder, the detrusor muscle contracts partially and prematurely as the bladder fills. The result, a sensation of urgency or an urge incontinence episode. The mechanisms that generate the feeling of urgency are the subject of much debate. One theory proposes that the parasympathetic nerves asynchronously release acetylcholine. Scattered acetylcholine binds to the M3 receptors, causing small, isolated contractions in the detrusor muscle as the bladder fills. If the micromotion of the detrusor muscle exceeds normal baseline tone, A-delta sensory nerves become overactivated. These excessive afferent signals may contribute to the sensation of urgency or leakage. Another theory claims that during bladder filling, as the urothelium stretches, it releases several agents, including ATP, acetylcholine, and nitric oxide. These agents may activate unmyelinated sensory C fibers, increasing the level of afferent signaling, which may generate the sensation of urgency. Regardless of the mechanism, it is clear that detrusor contraction plays a key role. Research on the contribution of muscarinic receptors, particularly of the M3 subtype, will help further our understanding of this debilitating condition.